You want to wait for 2-3 minutes?
I think we can start because it will be recorded, so people get to
Okay.
watch. Thank you.
Good morning. Can everyone see me and hear me?
Namaskar, Dr. Dinesh ji. He's my classmate.
Namaste, sir.
ठीक है.
Okay.
Can go ahead.
So good morning, everyone. Today we are again here with the GCAR Journal Club meeting. As we all know that Journal Club is growing and we are excited to have you on this journey with us. With over 1,200 YouTube subscribers and 2,000 newsletter readers, our community is passionate about advancing in Ayurveda, research and making scientific knowledge more accessible. We are looking for enthusiastic volunteers to join our team. Please support us. Volunteering with us is a great way to learn, connect with like minded individuals, and contribute to meaningful scientific communication. If
Would
you
you like to
enjoy
share
what
the
we
slide? Dr. Archana, would you like to share the slide?
Sure.
Thank you.
1 minute. I'm having a little issue with this thing.
No problem.
Just opened 1 minute because it does not let me go down to the vision and Dr. Pratibha, do you have the vision and mission slide? Because somehow it's like it's not letting me minimize the screen. Oh, wait. Yes, I'm sorry. Can everyone see my screen?
It's still loading.
Okay, something with my
Uh,
internet.
it's showing some, it's not showing the slide. Okay. Okay.
Is it now? So
Yeah.
our GCAR's mission is to encourage, educate, and facilitate basic clinical and interdisciplinary research in Ayurveda. Our vision is to promote and establish Ayurveda as an evidence-based health science globally. So we encourage all the volunteers to participate, to like, subscribe, and increase our YouTube subscribers. And please sign up for the newsletters as well. So we have our ongoing beautiful Dr. Pratibha, who is my mentor as well and who is like passionate for promoting Ayurveda. She has her degree in BMS, MD-Ayurveda, MPH. She's an internationally renowned Ayurveda expert. She's a senior faculty at several Ayurveda schools, CEO of My Ayurveda LLC and founder, president of two non-profits, Council for Ayurveda Research and Wholistic Health Alliance. She currently practices in the Greater Boston with a worldwide clientele. You can read more about her on our social media platforms. So I will hand over the stage to Dr. Pratibha and she can go ahead. Thank you.
Thank you, Dr. Archana. If you want to stop sharing, I can bring up my slides. Thank you so much. And I know we had a lot of registrations, but it's timing difference in the two countries. So I think people prefer to watch online later on. Okay. All right, welcome everyone to the Journal Club meeting for August. We had a speaker situation, so I have stepped in, and I'm very happy to be here to share about a publication and review it critically with all of you. And because I am this is kind of my baby in a way, this nonprofit and journal club. We have been doing this for so many months, years actually, that I would also take this opportunity, with your permission, to introduce some basic understanding of what a journal club is. And also later on, when I'm talking about the study, I will also share a little bit of meaning of some of the key terms that this study has mentioned. so that we are kind of keeping the educational part of it as alive as much as possible. So let's get started. This presentation is meant for educational purposes only. None of the information in this presentation is meant to replace a medical advice. The information presented in the slides is not verified or approved by FDA or any such agency. The presenter makes no claim, that's me, makes no claim, promise, or guarantee of any kind regarding the information presented here, and expressly disclaims liability of any kind. So there's just a something I need to read before presenting. Now, let's get started with what is a journal club before I actually go to the published article and start reviewing it with all of you with critical eyes. Let's just do a quick review of what a journal club is. So, it is an educational meeting in which a group of individuals discuss published articles to keep themselves abreast of new knowledge, promoting in them the awareness of current research findings, teaching them to critique and appraise research, and encourage them to utilize research in evidence based practice of the specialty, because there are millions of publications out there, but not all of them have the rigor of proper scientific study design or sample or bias, etc. So it is really important to engage in journal club meetings on a regular basis to continue to hone and sharpen the critical eye, to be able to discern between robust studies, well done and well published studies, versus poorly conducted as well as published studies. So let's keep that in mind. What is a journal club and what is the purpose of a journal club? It is a group of professionals meeting regularly, just like we are meeting right now, to critically discuss and appraise research articles, aiming to improve knowledge. What is the aim? To improve knowledge, to develop critical appraisal skills, and bridge the gap between research and practice to enhance patient care. Ultimate goal is to enhance patient care. And if we are able to utilize well done and well published research, then it enhances patient outcomes. So that is the ultimate goal. In the journal club, members typically take turns presenting, which is what we have been doing, and leading discussions on a recently published paper or an older paper, covering its background, methodology, findings, and clinical relevance. So you can take any paper, recent or old, and see how critically you have looked at the paper. That is the goal. Are you just taking the publication as is, the conclusions, the results, etc., or are you able to critically think about it, read about it, and then take away, make your own conclusions if the study is robust or not? So here members typically take turns in presenting about the paper. The primary goal is to foster a safe learning environment. So basically, this is a learning opportunity. There is nothing, me presenting, you listening. It's all of us participating in a learning environment where you can ask questions freely and also share perspectives without fear of ridicule. So I also hope you have had a chance to look at the paper so that you can also chime in towards the end. So I'll try to cover the presentation in 45, 50 minutes so that we can have a chance for some interaction. So, what are the key characteristics of a journal club? Critical appraisal. Participants analyze a research paper's strengths, weaknesses, and conclusions, focusing on the quality of data and methods. Knowledge sharing. Members gain exposure to new research, especially in rapidly evolving fields, and learn from each other's perspectives and expertise. Skill development. This is so important because we should not just become complacent in whatever our training has been, and not engage in further skill development activities. So this journal club also helps us to continue to develop new skills, which could be critical thinking, analytical thinking, communication skills, and more. Regularity. Now, journal clubs meet on a set schedule, so we meet monthly, for example. Rotating presentations, everybody gets a chance, so everybody gets to be an expert on a particular paper. So the goals are to improve patient outcomes, facilitate evidence-based practice, enhance professional education, and foster a collaborative atmosphere. So these slides will be available later on in our YouTube channel, so you can read in more detail there. What is the format of a journal club? Very important. A journal club runs on the IMRAD format, which stands for Introduction, Methods, Results, and Discussion. This is the acronym. This is the standard organizational structure for most scientific research articles, and it provides a logical framework for presenting and discussing a study. So, this framework is really um, perfect for presenting a paper, a published paper, because it allows you to go over all sections in a methodological manner and giving clear, concise information from the published paper. So here is a breakdown very quickly. Introduction. This explains the research question, provides background information on the topic, and justifies the importance of the study. A study has to be justified. There has to be a proper objective. The journal club presenter should cover the purpose, hypothesis, and significance of the research in the introduction section. Then comes the method section, in which how the study was conducted, including the study design, population, procedures, and data analysis. These are covered. During the club meeting, attendees will evaluate whether the study's design was appropriate and whether the results can be trusted. Then the results section has to be presented, which is typically done through figures, tables, and a concise summary of the data. The presenter should walk the group through key outcomes and highlight the significant trends. Then there is a discussion section where the data and the results interpretation is done and their meaning is explained in the context of existing literature and original hypothesis. The reference point is the original hypothesis. So this section is where the group will discuss the study's strengths, limitations, clinical relevance, and directions for future research. So now we come to the study that I have selected for today's Journal Club meeting. It is titled Efficacy and Safety of Shatavari Root Extract for the Management of Menopausal Symptoms, a double-blind, multicenter, randomized controlled trial. This is the title of the study. This article was published in Curious, which is an open access online medical journal that publishes peer reviewed research articles. It was published on April 8th, 2024. Therefore, it is a very recent article. The authors, Vani S. Gudise and Meenakshi P. Dasari, seem to be doctors in the obstetrics and gynecology department in Good Life Hospital, while the third principal investigator, Siva Sai K. Kuri Chetty, is from Rasastra and Vaishya Kalpana department at Sri Jeevana Ayurveda College. And all of these institutions are located in Hyderabad, India. Okay. So now let's start with the synopsis of the abstract. This will just give you a preview of the entire study. So the background is that that pre and post menopausal symptoms may impact the quality of life of menopausal women. Quality of life, we will shorten it as QOL. Hormone replacement therapy, HRT, which is the standard of care, may cause various side effects, some of which can be serious. This is driving women to seek alternative therapies for more customized care with less or no side effects. This particular study aimed to evaluate the safety and efficacy of the Ayurvedic herb Shatavari, which is a known and traditionally used herb for menopausal symptoms in comparison to a placebo. In this particular study, there was a placebo group as well. Methodology was a randomized, double-blinded, multi-center, placebo-controlled clinical study, which is the gold standard actually of clinical studies. A total of 70 patients were randomized to two groups. One, the test group or the active group, and then the second was the placebo group, where microcrystalline cellulose tablets were given with 35 participants in each group. The study outcome showed a positive and significant effect of the active test ingredient over the placebo in terms of reduction in hot flashes, night sweats, insomnia, anxiety, nervousness, vaginal dryness, and loss of libido, which are some of the key signs and symptoms in menopause. The UTN-QOL questionnaire. So UTN-QOL or quality of life questionnaire is specific to menopausal women. Otherwise, there are many other quality of life questionnaires available which are standardized. But UTN-QOL is specific to menopausal women. So the UTN-QOL improved significantly in the test group compared to the placebo group. No significant adverse events were recorded in the test group, suggesting the safety of this formulation. So there were two objectives, efficacy and safety, and both were verified in this clinical trial. The conclusions were the test compound could be a safe alternative to modern drugs. The findings of this study support the traditional use of Shatavari in Ayurveda. Further clinical and pharmacological studies with longer duration and larger and more diverse sample sizes are required to understand the generalized effect of Shatavari root extract in menopausal women. The keywords here mentioned in the study are Regensburg Insomnia Scale, UTN Quality of Life, Menopausal Symptoms, Shatavari, Ayurveda, Asparagus racemosus, menopause. These are some of the keywords with which the study can be searched on Google. So um, let's go on. Yeah, what I wanted to mention here was that when I started to read the study, one of the first thing that struck me was the small sample size. 70 patients is a small sample size. So we will see how the authors have talked about the limitations of the study, which is a very important part of a good study, that limitations are mentioned and small sample size is mentioned as a limitation. You will see later on. So that is a point in favor of this study. It's a good study. So, some of the meanings I wanted to share, if, in case some of you are new to how to read the studies. Multi-center is a word you have heard, which means that the trial or the study took place at multiple locations or research centers, increasing the diversity and generalizability of the results. So, it's always a good point if you see multi-center, because if it's done only at one center, those results could be specific to that particular population only. So, more centers, more robust the data can be. Randomization. So, participants were randomly assigned. So, usually randomized control trials have computer generated randomized assigning to the groups, whatever, one, two, not one, two, three, whatever groups might be there, uh, and controlled. So there is always a control group, meaning to say there will be one group that is used as a control in which medicine is not given. And that will be the comparison group, that for the same duration, for the same set of variables, one is given the actual intervention, the other is given something that looks like that, but is not an actual intervention. Double blind. This is really, really important to understand. In double blind studies, neither the study participants nor the researchers, PIs, they know what is being given to which study group. There is a assigned person, only that assigned person knows which one is a real medicine and which one is not. And those results are recorded with codes usually, which are not visible to the researcher or the participants. So, till the end, the participants will not know if they have received the intervention or the placebo. Okay, so this prevents, why is this done? This prevents bias from influencing the results. Okay, if the, for example, if the researcher knows this is the group that is getting the medication or the intervention, they might at follow up try to see some positive changes. So that bias is eliminated when the study is double blinded. This type of trial is considered the gold standard in clinical research because it minimizes bias and provides the strongest evidence for treatment's effectiveness and safety. So now, what is the purpose? Now, if you remember IMRAD, in the introduction, we have to talk about purpose, significance, and hypothesis of the study. So let's talk about it. So, menopause is defined as the permanent cessation of menstruation, typically occurring at the age of around the age of 50, plus or minus. It is diagnosed when a woman has not had a menstrual period for 12 consecutive months while not being pregnant, because pregnancy can also cause amenorrhea, or using hormonal contraceptives. So none of those reasons are present, and there has been 12 consecutive months of no menstrual period. Then a woman is said to be in menopause. Menopause is a pivotal, natural, and inevitable physiological milestone in the lives of women. The global population of postmenopausal women is on the rise. So here is the significance part of it, of the study. Why is the study significant? Because the global population of postmenopausal women is on the rise, constituting 26% of all females under the age of 50. Notably, there is a 21% increase in the occurrence of early age menopause. So, we are talking about an increasing population of menopausal women. And by 2025, there'll be, which is the year we are in, there will be approximately a little more than 1 billion menopausal women globally, about 1/8 of the Earth's population. Also, menopause is accompanied by multi-systemic symptoms such as hot flashes, vaginal dryness, disturbed sleep, insomnia, urogenital infections, osteoporosis, early onset of coronary heart disease, anxiety, depression, diabetes, cognitive difficulties. It's like a tsunami of conditions in menopause, all due to significant changes in hormonal levels, especially estrogen and progesterone. So, the quality of life is majorly impacted, and we are talking about 1 billion women. Their quality of life, which means their productivity, their participation in social and community work is diminished, So this is a very good reason for undertaking a study like this, where a better solution is being sought for resolving all of these signs and symptoms that menopausal women can experience with no or few side effects. That is the goal. So, approximately 75% of women experience perimenopausal or menopausal symptoms due to estrogen deficiency, and about 25% may not experience any signs or symptoms, but 75% women do. The transition from a perimenopausal state to menopause is often troublesome, mainly because of menopausal symptoms, and can affect the quality of life of these menopausal women. So, menopausal symptoms may be mitigated to a certain degree using local or systemic exogenous estrogen administration. Exogenous, which is given from outside, not generated within the body, via hormone replacement therapy. However, the use of HRT is linked with significant risks. Health risks, these have been linked to increased risks of blood clots, strokes, and certain types of cancers, particularly with long term use. The significance, so there are some references here you can read. Unsuitability for some women. Many women cannot take HRT due to pre existing conditions like a history of breast cancer, heart disease. So, if they have a history of breast cancer, which is ER positive, they cannot take hormone replacement therapy, heart disease, or blood clots because those can get aggravated with HRT. So, natural remedies provide a safer option for these individuals. And then there are other common side effects like bloating, breast tenderness, headaches, mood swings. So, there is desire to avoid these unpleasant symptoms um, and look for gentler alternatives. Here are some references from previous studies and surveys, which you can go over later. You can read the paper or you can read the slides in the on the YouTube channel. Here I have put two bullets in a different color, which I can explain now itself actually instead of later on. So in the next slide also you will see this black color bullet, which we can actually come back to later during interaction. I will mention why I put them in black color. So, Ayurveda offers a potential alternative to managing menopausal symptoms effectively and safely. This has led to an increased focus on exploring the effectiveness and safety of herbal and complementary therapies. Okay. Now, here the authors have mentioned that menopause can be studied under the concepts of Jara Vyadhi, Rajo Nivritti, Dhatukshaya, Vata Vriddhi, and other topics in Ayurvedic classics. The main principles of treatment are Rasayana Chikitsa and Pitta Vata Shamana. That's what the authors write in the paper. So, what are the benefits of a holistic patient centered approach? Effective natural remedies represent a holistic strategy that focuses on overall health, not just symptomatic alleviation, but overall well being, aligning with a more personalized and gentle approach to managing menopause, addressing diverse symptoms, not just specific symptoms that, okay, hot flashes and night sweats, I really need resolution, but other associated symptoms like vaginal dryness, insomnia, etc., mood swings, etc., can be addressed as well. Holistic approach also encompasses lifestyle changes advices, and therefore that can also be a positive aspect of a holistic patient-centered approach. Empowers patient choice. A wide variety of evidence-based natural options, including dietary changes and herbal supplements, offer women a choice in how they can manage their health. But there are also some considerations. So there could be variable efficacy and quality because all kinds of manufacturers are out there. Potential interactions and side effects. So there could be some interactions with other medications that the women could be on, and there could be some side effects. And you can see St. John's wort can have serious drug interactions, and black cohosh has been linked to liver issues in rare cases. There is need for medical supervision in herbal supplements, whereas a lot of them can be found over the counter. So these are some of the considerations. Uh, Now, what is the purpose and significance of the study? Again, continuing on that, Shatavari, which is an herb, has been widely used in Ayurveda for managing menopausal symptoms, but without much empirical evidence for effectiveness. So there are not many good studies out there that demonstrate in a certain sample size a consistent benefit. So that was one of the reasons this study was undertaken. Shatavari predominantly balances Pitta dosha followed by Vata dosha. This is what is mentioned in the study. And then they say Pitta dosha is the biological energy responsible for digestion, balancing, etc. So, Shatavari contains phytoestrogens, compounds known for their estrogen like effects, which play a crucial role in alleviating the discomfort associated with menopause. Steroidal saponins, these are the active principles. They're also called shatavarins. These are the principal bioactive constituents of shatavari root. There are other alkaloids, quercetin and glycosides of quercetin, etc. So, the traditional use of Shatavari suggests potential benefits in managing women's hormonal disorders, making it a noteworthy candidate for further investigation and research. Here, the test product. Now, this is important to note. The test product is something called Aspirus, which is a standardized herbal root extract of Shatavari. So, we will make a mental note that this is not a classical intervention. Rather, it is something that is used in these studies because it is possible to standardize it. So that is the reason we are talking about a herbal root extract. This clinical study was conducted to assess the safety and efficacy of the extract in the management of menopausal symptoms in humans. Okay, now what was the methodology? Okay, this was an 8-week multi-center interventional prospective, means ongoing, not retrospective, prospective. These are the terms to look up and understand very clearly. It was randomized, double-blind, placebo-controlled, parallel clinical trial structured to compare the safety and efficacy of the test active ingredient, which we saw was Aspirus. Okay, Aspirus, we'll remember that. Uh, with the placebo in the management of menopausal symptoms and regulation of the HPO axis, hypothalamus pituitary ovarian axis in pre and postmenopausal women. So we'll see the figure 1 now. The trials were conducted at 2 locations. So multi-center here is referring to just 2 locations. Okay, not too many, but just 2. The study was conducted in Good Life Hospital in Vijayawada. Here, they got 40 from the 70 in the sample. 40 were from Good Life Hospital in Vijayawada and 30 were from Ravi Nursing Home in Guntur, Andhra Pradesh, from August 10, 2023 to October 26, 2023. So this was the time of um, patient accrual. Okay. Now, this is a diagram. It says consort flow chart. What is a consort chart? Let's first understand that. A consort chart is a consolidated standards of reporting trials flow diagram. This is a visual tool used in clinical research, particularly in RCTs, to track the movement of participants through the study's various phases. Okay, from initial screening and enrollment to random allocation, intervention assignment, follow up, and final analysis. So these diagrams provide transparency and details by showing the number of participants at each stage and the reasons why participants were excluded or dropped out, helping readers assess the validity and the generalizability of the trial's findings. Okay. So this is the consort flow diagram for this study. So enrollment, they were assessed for eligibility and n is equal to n is the number usually, n means number. So 70 participants were found to be eligible and we'll see what was the eligibility criteria or the inclusion criteria. So excluded, none. None were excluded. Not meeting inclusion criteria, none. Declined to participate, none. Other reasons, none. So that's clear. Randomized, 70 participants. Allocation, baseline, day 0, allocated to intervention, N35. So 35 participants were allocated to medication group and received allocated intervention 250 milligram vegetable capsules of Aspirus for 35 participants. And on the right side, you see another 35 participants who were allocated to the placebo group who received the crystalline salt as an intervention. That's a placebo, basically. Uh, and then follow-up was done on day 7, day 14, day 30, and day 60. Lost to follow-up in the intervention group, 0. None lost to follow-up, no attrition at all. Discontinued intervention, 0. None. Lost to follow-up in the placebo group, 2. Discontinued intervention, 0. Analysis, Full 35 were analyzed in the intervention arm, and in the placebo arm, 33 were analyzed because two were lost to follow up. Why they were lost to follow up? I believe that detail is not given. Okay. So, methods again, under the methods, we have to think about ethical considerations. So, IRB approval or human independent ethics committee approval was obtained as per the declarations of Helsinki from Fusion Clinical Research, Vijayawada, which must be an authorized agency for. Um, I think here we call it IRB. I think in India it's called a little bit different, but human trial in permission, basically consent. The test product description, let's understand what was that. So the product used was Aspirus, which is a full spectrum Shatavari extract that focuses on delivering a standardized Asparagus racemosus root extract. The extraction was done using a unique proprietary method, and the root extract was standardized to the phytoconstituents of total shatavarins of 5% with high performance thin layered chromatography. The allergen free test product was tested for heavy metals such as lead, arsenic, cadmium, and mercury, following the Association of Official Agricultural Chemists standard methodology. So they followed protocol here, proper protocol. What was the study protocol and what was the participation selection criteria? This is a very important part of any study, that the selection criteria has to be mentioned and explained very clearly. So the inclusion criteria or the selection criteria here was pre and post menopausal women. This is important, not just pre or not just post, but pre and post menopausal women aged between 40 and 65 years who were experiencing menopausal symptoms such as hot flashes, night sweats, anxiety, fatigue, depression, insomnia, mood swings, as diagnosed by the study investigator or investigators. The exclusion criteria were pregnant women or lactating women. So even if they had all these symptoms and they were in the age group, but if they were pregnant or if they were lactating or who were actively trying to conceive, participants who had undergone hysterectomy, and women who were on HRT, hormone replacement therapy, or any other herbal treatments for the management of menopausal symptoms, these women were not included. Okay, this is called exclusion criteria. Participants with a history of any serious illnesses in the last 3 months or any history of current alcohol or substance abuse were also excluded. Other exclusion criteria included women with an allergy or sensitivity to any ingredients in the study treatment, those who had participated in any other clinical studies in the past 3 months, because those interventions could still be there in their system, or those who intended to participate in other clinical studies parallelly, because there would be um, um, interference with the intervention for this study, right? I hope this is clear. And this is really important because inclusion criteria has to be defined very, very clearly. But not everybody who qualifies for those selection or inclusion criteria will make it in the study, because there are some exclusion criteria as well. Okay, so both are very important, and they were very clearly defined in this study. A total of 70 women who met all the inclusion and exclusion criteria and provided informed consent were enrolled in the trial. They were randomly assigned to either the active or placebo group using a computer generated schedule. All recruited women were administered one capsule of 250 milligram containing either the active Shatavari root extract or placebo, and these were given twice a day for 60 days, 8 weeks. Detailed demographic, medical, and medication histories were obtained from the participants. Vital parameters were collected on day 1, day 30, and day 60. So, parameters were collected only on 3 visits: one baseline, day 1, and then 2 follow up visits. Even though, if you remember the uh, where is that? The consort flow chart. There were follow-ups on 7, 14, 30, and 60. So there were five total visits, okay? Five total visits, but vitals were collected only on day one with baseline data, day 30, and day 60. This is what you're going to see in the tables and figures. So we'll remember this, okay? Laboratory results tests to determine the levels of serum estradiol and progesterone were also performed. So these are quantitative data. These two are quantitative data, whereas most of the other things that menopausal syndrome entails are qualitative data, right? Hot flashes, night sweats, you can't quantify, it's a qualitative um, reporting. So, what were the outcome measures? Primary outcome measures, the changes in the, these are also important. The outcome measures have to be clearly defined and described and listed. The changes, so what were we looking for in the study? If I'm a principal investigator, I'm looking for a change in the UTN quality of life scores. Okay, if they were low before the study, have they improved? That is what I want to look at. And there are some secondary outcome measures listed as well. So, primary is the total UTN quality of life scores on day 30 and day 60 compared to the baseline scores. I hope this is clear to everyone. And the secondary outcome measures were evaluation of the changes in specific parameters such as individual domain scores of UQOL, UTN quality of life, which were specifically occupational related, health related, emotional related, and sexual related. Okay. The score, and then score of all three: depression, anxiety, and stress, domains of depression and anxiety stress scale, DAS. It's called DAS scale, which has 21 questions. That was a secondary outcome measure. The total score of Regensburg insomnia scale, which measures insomnia, poor sleep depth, poor sleep quality, fearful focus on insomnia, all these questions. Okay. Menopausal symptoms using a 5-point Likert scale. Okay, Likert scale, I hope this is familiar to everybody. Like we say, on a scale of 1 to 5, how do you feel today? That is Likert scale. So this 5-point Likert scale is listed as a secondary outcome measure for menopausal symptoms. Okay, so for example, hot flashes on a scale of 1 to 5, where are your hot flashes today compared to the 4 that you reported at baseline, like that. Then serum estradiol and progesterone were measured at these three stages. And then, interestingly, they also did a treatment satisfaction questionnaire, which is not related to the intervention at all, but they did it as a separate additional outcome measure at 30-day and at 60 days. So, changes in any vital parameters, pulse, blood pressure, respiration, etc., were also examined. Safety outcomes. Safety of the trial intervention was evaluated by recording the incidence of adverse events on every scheduled follow-up visit. All adverse events during the study timeline were recorded and monitored following the good clinical practice guidelines. You can look these up to understand them better. I'm trying to keep an eye on the time. So, statistical analysis, I'm not going to go into detail because you have to be a statistician to understand these methods that were applied. You can open the study itself and read in detail and understand each and every test that was applied to different conditions and situations and get a better sense of it. What is most important is that the statistical significance has to be below the p-value. You know, the p-value that we talk about should be below 0.05. Only then it is statistically significant, which you will see there was a lot of statistically significant outcomes. Okay. So this I will let you go over. Results. Now let's talk about results. So I have just taken the figures and the tables. You can see I just have time to take you over the figures, not the tables. But you will understand. Now, here you have to see the legend on the right side. Red is for placebo and blue is for active intervention. So, placebo, we have already seen that two people attrition happened. So, n is equal to 33, active n is equal to 35, full participants who were enrolled. So, you can see that their quality of life at baseline is similar. They're both groups, women are in the same boat. They're having similar level of issues. Their quality of life scores are similarly poor. However, in the blue group, which is the intervention group, at 30 day, there is beginning to be improvement. And at 60 days, the improvement has increased. Okay, I hope this is clear. And here you can see the actual table, and you can see that the p value, you can see less than 0.0001, which is very, very significant. Okay, so the p value is very significant. So these are significant changes. Again, I'm going to be able to take you only over the figures, not the tables, because of lack of time. Um, you can see the baseline data here, and at 30 days and 60 days, depression, anxiety, and stress are significantly reduced. You can see, right? In the blue, you can see the bars are lower. Okay. Similarly, here you can see all the results are statistically very, very significant. P value is 0.0001. Here, this is the insomnia scale. Here also you can see consistent improvement. Consistent improvement. You see the blue is coming down at 30 days and then more lower at 60 days, which is a good thing, which means all the problems related with sleep are reducing. Okay. Now, this is a Likert scale for all the qualitative symptoms like mood swings. You can see for placebo how it is, and for aspirus how it is. It's reducing. Okay. So I will let you go over these figures in detail at your end, but you can see that there is a consistent improvement in all parameters. So, you can see that to understand the test product on menopausal symptoms, measures such as the total UQOL, which is 14, and DAS 21, these are just references, sorry. These are just references which I have not deleted. So DAS 20, these are the scales that were used. And the results of this study showed that the subjects in the active group had a significant change in vasomotor symptoms with reduced daily hot flashes, night sweats, anxiety, and depression, along with improvement in physical, mental, and sexual health care criteria. The vitals were unchanged when reported pre and post supplementation of the formulation. So vitals, whatever BP, pulse, etc., they did not change. The primary outcome measure, changes in the total UQL, was better in the active group compared to the placebo group. Middle-aged women were considered as the ideal study participants in this study. In many subjects, QOL scores were suboptimal to begin with, and these are because if you look at the bullet number 5, these can be attributed to personal circumstances of women such as unemployment, problems in managing children, interpersonal relationships, family issues. So poor QL to begin with, but with the intervention, those QLs improved. And all across emotional, sexual, occupational, so different types of questions, specific questions under the UT and QOL were assessed, and all showed significant better outcome. Okay. So this also is talking about the phytoestrogen in the intervention product. Okay. So I'm going to rush through these slides, which you can also look up in the study. Uh, so again, serum estradiol and progesterone were also significantly improved in the active group. Um, So now study limitations. So overall in the discussion section, the summary of that is that consistent and progressive improvements were noticed across all of the primary as well as the secondary outcomes that were determined at the beginning of the study itself. Now, a good study should always mention limitations. And this study does mention limitations clearly. A small sample size with specific age groups, participants with varying stages of severity in symptoms. So there was not enough uniformity there. Fluctuating estradiol levels of the study participants, which is natural. So, did that affect the outcomes? Those were the limitations because these may mask some of the study outcomes. Hence, a larger sample size, more centers of study with more diverse samples and inclusion of diverse age groups may help generalize and confirm the present observations. Okay. Um, I also want to quickly go to the black part that I had mentioned, black lining, these. So this and this. The point that I wanted to share with all of you is they have mentioned Ayurvedic terminologies without giving their meaning anywhere. I would have loved to see an appendix where these terms are explained and also more about the product, who um, made that standardized product, which was the company, what is their background. Those details are not available. So I would add that to the limitations from my end. Um, Okay, what were the conclusions? Most studies conducted so far are on postmenopausal women, whereas this study also included premenopausal women. Moreover, very few studies have shown the safety and efficacy of Shatavari alone on humans. The findings from this study support the positive effects of a novel Shatavari formulation taken for 60 days at a dose of 250 milligram twice a day. This study also showed that the test product was safe and well tolerated with no major adverse events in the participants with menopausal symptoms. Those are very well documented in the tables. So please have a look. This study is actually very interesting. So I would really encourage everybody to sit down and read the study. It's a good example or template or model for how to write up a study once it has. So conducting a good study is one thing, and then writing it up for um, good scientific feedback and application into patient care is another thing. So both have to be done well and with proper oversight, effort, and diligence. So I give this study high marks for that reason on my scale. Okay, so therefore, it could be a safe alternative to modern drugs. The efficacy of test product was well demonstrated. The findings of this study support the traditional use of Shatavari with modern scientific evidence. Further clinical and pharmacological studies with longer durations and larger sample sizes are required to understand the mechanistic actions of this formulation in menopausal women. They also obtained human ethics consent, which I mentioned in the beginning, but also they additionally confirmed that this study did not involve animal subjects or tissue. Okay, so we have exactly 5 minutes for discussions. So I would really request if you want to come on video, audio, and share your uh, impressions, thoughts. हाँ जी,
Good
डॉक्टर
evening,
दिनेश।
Dr. Pratibha.
हाँ जी, गुड इवनिंग।
I am Dr. Dinesh Rai. from Dr. Yasar, Rajasthan Ayurveda University, Jodhpur. Good evening, Dr. Pratibha. Your
जी जी
presentation is nice presentation.
थैंक यू
In my textbook, Ayurvedic textbook, Shatavari is the uterine tonic. It protects balance of the hormone and treat menopausal syndrome. There are so many clinical studies about the function of the Shatavari, as we very well know. In practice, we use Shatavari churn, Shatavari churn, Shatavari kalp, Shatavarix granules. So many products are produced in market. We generally them use in my practice. You present nice presentation. You describe how can we research and write good scientific paper. I appreciate her. Thank you. And very thank you.
thank you Dr. Dinesh. So I totally agree with you that we have so many traditional and classical Shatavari products that we use. But because this is a randomized controlled trial, a
Yes, yes,
standardized intervention is needed. And it is good to be able to measure up on Western scientific framework, while we also are, there is also work going on whole person research template. So we are
yes, yes,
trying to, there is an effort to try and develop a research template that is more uh, suitable for a holistic system like Ayurveda, because randomized controlled trials are not 100% suitable for a system like Ayurveda, because we like to use polyherbals and um, our authentic preparations, because extract is not something we mention in Ayurveda.
yes, yes. You are right.
Thank you. Thank you. Rutu, you want to come on audio video and tell us where you are joining us from?
Ritu, I
Yeah.
see your chat. So can you please?
Uh, yeah. Hello, Dr. Pratibha. I'm Dr. Ritu Sethi. I'm from Delhi. I'm HOD of Ayurveda Department in Holy Family Hospital, New Delhi.
Oh, wonderful, Dr. Ritu. Thank you for
Yeah,
joining.
yeah, yeah, yeah. So I was keen to, listen about your research. So it's actually amazing. We are using. I've done my MD in Kai Chikitsa from Gujarat Ayurveda University, and I'm
Wonderful.
practicing Ayurveda since 26 years. So
Uh,
post my MD. So you have done a great work, Dr. Pratibha. Congratulations, actually. So it's a beautiful work, and see, we all know you. We all are Ayurveda people, and we know about Shatavari, and we are using it since years together. And in many conditions, many in women, we have seen best of the best results, especially in fragile and 40 after 40 females in menopausal conditions. We have seen the results, but no one has proven this thing on scientific parameters like you have done. So it's really good.
Ritu, I must clarify, this is not my research.
Okay.
I'm just presenting a
Okay.
critical review
Ha
of
ha ha
a
ha.
published
All
paper,
right.
yeah,
Oh
in
wow.
India.
Acha.
Yes,
Okay.
yes,
Okay.
yes.
But
That
this
is
is
the purpose
amazing.
of Journal Club.
Yeah, yeah. This is amazing actually. The study is
Thank
really
you.
great, and
Thank
I would like to share this on other platforms also.
Thank you
So
so much.
that this work has been done, and this is already proven on scientific parameters.
Yes, and it is a
Yeah,
2024 study, so maybe a lot of people don't know about it,
yeah, might be
So,
certain.
um,
Hmm.
This is also needed. Sometimes we
Yeah,
need
yeah,
to
of
be
course.
able
Yeah,
to
yeah,
stand in scientific forums and talk with
of course.
authenticity. Here is
Yeah,
the
yeah,
data,
of
here is
course.
a study. It's a small sample
Mhm.
size of 70, but
Mhm.
still, it has shown enough meaningful outcome and result to
I think you are very humble to say that it is a small sample size. It is a very good sample size, actually.
70 is still considered small
Yeah,
on
yeah,
a
I understand.
scientific
Yeah, yeah, of
basis.
course. Yeah, yeah,
Yeah,
yeah, yeah.
but
Mhm.
it's very encouraging to
Yeah,
see
of course, definitely.
um,
Mhm.
these kind of outcomes, which are
Mhm.
properly documented.
Mhm.
Yeah,
Mhm. And even in lactating women, we all know that we have, Shatavrex granules. And I myself has once, when I was doing my post graduation, I made, Shatavari granules at my home, and I
right.
gave to some of my patients, and they were really very very enthusiastic that whatever the medicine you made that time, once you gave, which you made, you should give it to us repeatedly, because that was very very effective. Then because afterwards I was using the other prepared products from different pharmaceutical companies, but those I made and I gave to my patients, they were really got highly appreciated that time.
Wonderful.
So,
Wonderful. Thank you so much for
so
joining, everybody.
yeah,
Thank you so much for
yeah.
your inputs and interactions. And please visit our YouTube channel and you will find so many Journal Club presentations. We have about 1,200 subscribers right now. You
Great.
can listen to earlier presentations and recordings as well. And if you're interested in presenting, please reach out to us and we would love to have you present in our Journal Club meeting. Okay.
Thanks
Namaste.
a
Thank
lot,
you
Dr.
so much.
Pratibha. Namaskar to you.
Thank you. Thank you. Dr. Archana, you
Okay.
want to conclude?
Yeah, thank you so much. Thank you, Dr. Pratibha, for giving us another insight on use of Shatavari. I know everybody knows Shatavari, but how effective the paper were presented and what were the shortcoming in the paper. And I hope like in future, all the researchers will take all this into consideration and present another good scientific approach towards an scientific paper where people are getting benefited. And I agree with Dr. Ritu, like Shatavarix granules has benefits on the lactating mothers, and I personally have used it and experienced it. So how powerful and potent Shatavari is, we all know. And through your presentation, we also came to know another aspect of that, how beneficial and productive
Of
and
course.
potent it can be. So we are looking more to learn from you, and thank you so much everyone who attended. And please do subscribe to our YouTube and our newsletter. We want more volunteers to participate. And anyone who's interested, just email us or just reach out to Dr. Pratibha or anyone who is like, or me, and we can definitely add you in the group. Thank you so much.
Thank you. Namaste.